Thank you very much for presenting that very challenging and visionary approach to data transparency. We're going to shift gears a little bit now and talk about opportunities and models that foster pooling and analysis of data. The first presenter is John Wagner from Merck, who's the Vice President and Head of Project Management and Early Development Projects.
Thanks very much, and to the organizers for the kind invitation to come here. The disclaimer is that these are personal impressions. I'm not representing the Biomarkers Consortium nor Merck, but most of what I'm talking about is actually published. Just a word about pre-competitive collaboration. The pre-competitive collaboration is something that the biomedical enterprise freely stole from the software industry. And why? Because it's something that probably works pretty well in increasing the efficiency of work. There's a great definition in this book by Steven Weber: competitors share early stages of research that benefit all. And I think there's a growing awareness, acknowledgement in the biomedical enterprise, in academics, government, and industry that pre-competitive collaboration works and can be used to really drive some work in our increasingly complex world. So the Biomarkers Consortium is a pre-competitive collaboration, and the nice thing about the consortium is that it's a group of institutions, companies, government agencies that have come together with a common goal for biomarkers. And there's a really key role in having our regulatory colleagues at the table, especially for biomarkers. Biomarkers are going to be used more broadly in various possibly regulatory sorts of situations. So the goals of the Biomarkers Consortium were laid out as facilitating the development of new biomarkers, qualifying those biomarkers for use, generating information that's useful, possibly to inform regulatory decision making, and then importantly making all of this completely public. I want to talk just briefly about one biomarker, adiponectin. And this is a protein biomarker that was discovered in the 1990s, and it's associated with there's decreases in adiponectin in obesity, decreases in insulin sensitivity, and drugs like pioglitazone, PPAR agonists, increase adiponectin. Adiponectin is thought to possibly be part of the mechanism for insulin resistance, and also part of the mechanism by which PPAR drugs like pioglitazone exert their effects. It's very clear that these medications drive up adiponectin concentrations very quickly in patients, in healthy volunteers, and in patients. Now, the issue with adiponectin when this project was conceived is that there was quite a bit of research on adiponectin, these associations with insulin resistance. But one question that wasn't addressed is whether adiponectin, a response in this biomarker adiponectin, could predict a hemoglobin A1C decrease in patients with type 2 diabetes treated with drugs like pioglitazone. Why is that important? Well, if there were a good predictive biomarker like that, it could be used for patient segmentation, and the patients who were not going to respond to these agents could get other therapies, and it would only be the patients who were likely to respond that could get these medications. Now, it's interesting that this was a very active area of research, and there were many file drawers and computer hard drives full of adiponectin data in various companies and in academic institutions. But the questions, the data that addressed this question were relatively sparse data. that were publicly available. And clinicaltrials.gov doesn't answer this because most of these data were older and also were exploratory endpoints, at least the adiponectin part, so not really readily available, at least in any kind of an easy way. So when the biomarkers consortium was put together, there was this was really a proof of concept project. This was thought to be something that would be relatively simple to put all this data together and answer this important research question. So a number of companies did agree, GSK, Lilly, Merck, Roche, to put together the data, actually a blinded data set for many trials in over 2,000 patients. And we had statistical analysis that was donated by NIH and by Quintiles. And this project went through a series of phases. I won't describe all of it. The first two phases were really meant to assess whether we had a good database, whether it was valid for answering the questions that we really wanted to get to. And it was the last phase of the research where we were asking the question about the prognostic value of adiponectin and hemoglobin A1C. We worked through a project team and then made those results publicly available very quickly. So this is basically the result of all of that work, and it turns out that adiponectin is correlated with hemoglobin A1C lowering. That correlation isn't as good as fasting plasma glucose. That's not really an enormous surprise, but it's still a very important observation because the fasting glucose changes occur very late in treatment, and the adiponectin changes occur relatively early. So even with a modest correlation like this, there was really a hope that this could be used as a predictive biomarker. And that's shown in a receiver-operator curve kind of a model in which the receiver-operator curve characteristics of fasting plasma glucose and adiponectin are pretty similar. So we concluded that adiponectin is a robust predictor of glycemic response to these PPAR agents. We confirmed a lot of previous findings in this aggregated database and really concluded that the potential utility of adiponectin across the spectrum of glucose tolerance was well demonstrated. I think for this audience, the most important conclusion was that this project established that a cross-company collaboration was a robust and feasible method for doing this kind of research. And we kept this project team going even after the result in order to really delve into some of the lessons learned. So transparently, this project, this is the timeline for the project. And you can see that it took a long time to accomplish this project. All of that data was collected well before 2007. In fact, the kernel for this project was really conceived of several years before the institution of the Biomarkers Consortium prior to 2005. And so the time it took to really accomplish this is pretty long, and that's a real problem. You can see that there's many steps along the way. As we learned about how to collaborate better, this was one of the initial projects. It was the first published project by the Biomarkers Consortium. We were getting better at it, but it did take a long time to accomplish. So we learned many lessons in this consortium. One of the issues was that the focus, organization, and pace of the group really sunk to the lowest common denominator. Now, this is a group of busy industry scientists, some collaborators in a CRO, collaborators in the NIH, and it was not always the first priority of everyone involved in the project. You have to be honest. It was the evening job for many people, myself included. And that's really a problem. We addressed that in part by making sure that we used good project management skills, and in fact this became a best practice at the Biomarkers Consortium of having a very good project manager associated with all the projects. There also was a lack of collaboration tools initially, which the Biomarkers Consortium has also addressed. We really struggled with the data sharing principles and standards. It was difficult to negotiate the data sharing plan with even the small set of companies to a level that satisfied everybody. The definitions, the standard definitions, were not all obvious. And someone else mentioned it earlier, some people left particular companies who actually had some of the crucial information. So there was limited institutional memory. It was incredibly important for this project to have a single knowledgeable legal liaison who would be responsible at the individual companies. In fact, the knowledgeable part was really important because a lot of legal folks in large companies, this data sharing is not their day job either, and we really had to spend a lot of time explaining what the purpose of this project was. In the end, this project came to a template for the Biomarkers Consortium for Data Sharing Plans, which is hopefully driving faster work for future projects. There also was a lot of issues with the limitations of the existing data. This was an existing data sharing project. It wasn't conceived of, the studies that we were using weren't conceived to answer this research question, so there were real limitations. There also were limitations in the adiponectin data itself, because different companies use different assays, and we had to do some work to assure ourselves that we could really combine that data. For projects like this, we really just need to acknowledge the limitations and move on, and have prospective follow-up when that's necessary. Now, there's also some broader issues that we ought to talk about. Some of them have already come up this morning. The clarity of the question, your research question, is really very important for defining the type of collaboration. The role of a neutral convener, in this case the Foundation for the NIH, was very important in gaining the trust of all of the folks who were, all of the stakeholders who were involved in this. Dialoguing with the FDA is incredibly important, and in this case we had an FDA representative, and we probably didn't even push that far enough in terms of making sure that we were qualifying this biomarker in a regulatory acceptable way. There were definitely behaviors that impeded this collaboration, not the least of which was just the time that the project team members could dedicate to this effort. And then some more general issues. We really observed that most collaborations, this one included, had even itself had a siloed attribute to it, and it was really incomplete. I need to point out that not all companies that had adiponectin data shared in this effort, and not all of the data, the adiponectin data in each company, could be merged into this data set. That's an issue. And the motivations were also an issue. Now, the Institute of Medicine had been involved in, through the National Cancer Policy Forum, an effort where really work some of the issues around the clarity of the question. And this is an article by Jill Altschuler that came out of some of those efforts. And we won't spend much time here, but if you look on the left-hand side there, it's boxes that represent how open the contributions are and how open the output is. And depending on the kind of question that you're asking, the openness of the contribution and the output are going to change. Also, the type of question, whether you're developing standards and tools, aggregating data, creating new knowledge, or developing a product, is going to change the kind of collaboration that you want to set up. The Biomarkers Consortium is is a consortium that's labeled number 4 in this slide. That is a restricted contribution. The parties that involved in the collaboration are named and restricted, but the output is completely open. And this is a good model for generating new knowledge. I think that the key role of the neutral convener is now readily acknowledged. The role of the FDA or other regulators is readily acknowledged. But some of the other issues here continue to be an impediment, in my opinion. We really need trust and openness when we're engaging in this kind of collaborative behavior. We can improve collaborations by really being very clear about our common goals and the unique value each party brings to bear. And we really need to strive for open inclusiveness in these appropriate collaborations. All the parties with the stakeholders with the data really need to be there. And this last point was also brought up earlier. You know, the motivations are similar and different across stakeholders. If we continue to reward academic folks with a sort of principal investigator laurels, and then reward pharmaceutical companies with for secrecy, then we're not going to get what we want in terms of these kinds of collaborations. So finally, I just want to acknowledge the folks who participated in this. On the left are all the folks who were involved in this Adiponectin project team that was through the Biomarkers Consortium, and on the right were folks who were involved in a pre-competitive collaboration workshop like this one that was convened by the National Cancer Policy Forum. Thanks.